Hello, welcome to week 4, amino acids and proteins. First, we're going to talk about a little bit of the basic chemistry behind proteins and amino acids. Amino acids are the organic compounds containing both an amino group, which looks like this, and the carboxyl group, which looks like this. Amino acids that occur in proteins are called alpha amino acids. We have a couple different ways to look at the formulas for these. The empirical formula just lists the chemical compounds, as you can see here. The core of the amino acid is the alpha carbon atom next to the carboxylic acid group. Here's a typical amino acid. They all look very similar in structure, but what makes them different from one another is the group that would be attached here, where you can see the R. Within the amino acids, we have something called a peptide bond. A peptide bond is a chemical bond formed between the carboxyl group of one amino acid and the amino group of the other. Amino acids combining in chains up to 5 in length are called oligopeptides. When we get longer chains, which could be 6 to 30 amino acids in a row, we call them polypeptides. And when we get more than 40 amino acids in a row, it constitutes a whole protein. So if you think about the steak that you eat, yes, it is protein. It consists of many, many amino acids put together in a certain way in order to create that. Kind of like things within our body. We eat amino acids in our foods. Our body is able to package those and turn them into other proteins within our body. When you think of protein in your body, a lot of you are probably thinking of your muscles and things like that, and that is true. But there are other proteins within our body that we need that all have different functions. So as we move through this chapter, we're going to talk about amino acids, amino acidopathies, which are issues and diseases you can have if there's a problem in the production of the amino acids making a protein. And then we're going to talk about the different proteins and what they do in your body. All right, so first looking at the amino acids, like we just showed you, there's one amino and one carboxylic acid functional group. We have a chain of amino acids, which could be a polypeptide, or greater than 40 again would be your protein. We have 10 amino acids that are needed by us humans and can be synthesized within the body. You can actually make them. There's 10 others that must be supplied within the proteins and the foods that we eat. Now, those are called essential amino acids. There are a list of them here, the ones that you have to have in your body, but you don't have to memorize this list. I just have it here just so you can kind of see what they look like. Now, they all end in "ine," which is kind of helps you to memorize that. If you see these words, you think it's an amino acid. Arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan. Well, tryptophan's kind of an exception, and valine. So the primary supply of amino acids for protein comes from our dietary protein. So if you eat that big steak, your body breaks it down into amino acids, which are then brought to your liver and packaged into another type of protein that you might need. Those are called essential amino acids, once again, the ones that you must get from your diet. They're essential for maintaining your health, growth. It's in the intestinal tract on indigested proteins. Those release the amino acids from the steak that you've just eaten, or chicken, or whatever type of protein it may be. And then those amino acids are absorbed from your jejunum in your intestine and into the blood, which enter your body's amino acid pool. Most of this process takes place within the liver. Once it's in the liver and other tissues draw from that amino acid pool and synthesize other proteins. The liver and kidneys also play a role in the excretion, so if you have too many, you're actually able to eliminate some of those by way of the urine in the kidneys. We have a couple different ways of using amino acids. One of them is called transamination. That's taking one amino acid and interconverting it into something else. Or deamination, pulling it apart or degrading it. The deamination produces ammonium ions, which are used in the synthesis of urea. Urea is the waste product that we find in the urine. So when you do have excess amino acids, you go to the big steakhouse where you eat the 48-ounce beef eater steak. If you have that situation where you're going to have way too much amino acids, your body's actually able to break those down, take the excess ammonium ions, and excrete them by way of urea in the kidneys. But don't worry, we have a whole NPN chapter coming up next week, and we'll go into more detail on that. So our amino acid concentrations vary during the day by 30%. I would imagine it goes up and down depending on your diet and what you're eating. We find that they're very high during the first 5 days of life, especially with premature neonates. And maternal values are low in the first half of pregnancy, probably because they're being used in the production of a fetus, I would imagine. Urine excretion varies with age as well. I would imagine as you get older and your kidney function decreases, you probably would not have the amino acid concentration in your urine per se as you would if you were younger. All right, now we're going to look at some amino acidopathies. These are some of the diseases involving amino acids that somebody could have. There's over 100 identified diseases. They're inherited. Usually there's some type of an enzyme missing, which is required for the production of a protein. So I do have a chart coming up here which will make it make a little more sense. So in here we have phenylalanine and tyrosine pathway. When we have phenylalanine in our body is usually able to convert it to tyrosine, which then converts it to hydroxyphenylpyruvic acid, which then converts it to homogenistic acid, which then converts it to malacetoacetate and fumarate acetoacetate. Now, where you see these red lines is where an enzyme is usually present to convert these chemicals to the next thing. Now, if you have something called PKU disorder, which we're probably going to talk about first here in a minute, what happens is you're missing this enzyme, phenylalanine hydroxylase. And if you don't have phenylalanine hydroxylase, you're not able to convert phenylalanine to tyrosine. So what happens is phenylalanine builds up in your brain and causes brain damage. The same thing can happen with tyrosine. You can be missing an enzyme to turn it into hydroxyphenylalanine. And if that happens, you end up with Tyrosinemia. Over here, homogenistic acid, if you don't have the enzyme to convert it to malacetoacetate, then you end up with a kind of a black colored urine, which we'll talk about here in a little bit. All right, so let's look at the first one, Phenylketonuria. This was the first one that I talked about in the chart above. It's an inherited autosomal recessive trait. It occurs in approximately 1 out of 15,000 births. It has almost a complete absence of that phenylalanine hydroxylase enzyme, which catalyzes the conversion of phenylalanine to tyrosine. So if I go back here, this enzyme right here is missing, so you cannot go from here to here. So this builds up. All right, so phenylpyruvic acid is a primary metabolite, but other compounds are seen as well. What's important to know is that if that phenylalanine builds up in your brain, it can cause deterioration of brain function, usually by the second or third week of life. It can be very damaging. In most states, it's required that screenings are done at birth so that we can prevent this disability. So I know my children, when they were born, they did a heel stick and they took all kinds of little blood samples on a piece of paper and they sent it to the state lab to test for these types of things. If they do find it early enough, they can provide a formula that does not have phenylalanine in it and prevent these types of issues from happening. A type of testing that is done is the Guthrie bacterial inhibition assay. Normal levels are usually 1.2 to 3.4 milligrams per deciliter. And we can also do prenatal diagnosis by DNA. And that's usually not something that would be done unless we know that the mom has had other children with this issue. The next one is Tyrosinemia. This is a familial metabolic disorder of tyrosine catabolism characterized by excretion of tyrosine and tyrosine catabolites in the urine. Elevated levels lead to liver damage, which may be fatal in infancy or cause liver cancer later on. I do have a video here that you can watch on your own time. It's about 8 minutes long. And if you want to go into your PowerPoint, you can see this. It's a little boy who was born with this, and the foods that they were giving him were making him very toxic. Once they switched him to other foods, it changed their life completely. Very interesting. I recommend you go back and watch this. Alkaptonuria, you can see the urine up on the top right hand side here, and this one is normal on the left. On the right would be the black urine that we would find with Alkaptonuria. With this one, it's very rare. It's 1 in 250,000 births. It's an accumulation of homogenistic acid which oxidizes to produce a black urine upon standing. So when you first get the urine in the lab, it's going to look like the one on the left. As it sits at room temperature, it will turn black. Patients usually don't have any immediate problems but can end up with arthritis later in life. My students memorize this one, black urine. Alkaptonuria sounds like Al Capone. He always wore those black suits. So they always say Al Capone, black suits, black urine. But you also have to know homogenistic acid too. Next one's really easy to remember. Maple syrup urine disease. With this one, the child will actually smell like burnt sugar or maple syrup, and especially the diaper. The diaper of the baby will smell like maple syrup. It results from an absence or reduced activity of branched chain alpha keto acid decarboxylase, which blocks the normal metabolism of the 3 essential amino acids, leucine, isoleucine, and valine. With this very low, 1 in 216,000 births, so similar to the Alkaptonuria, but the leucine levels are above 4. So my students always said, well, little Lucy is 4 years old and likes maple syrup. So if that helps you, go for it. Next one is Isovaleric Acidemia. This one produces a sweaty feet odor. People with sweaty feet that stink tend to be more isolated than other people, so my students always say Isovaleric Acidemia with the sweaty feet one. It can become very toxic to your central nervous system. You need to go on a protein restrictive diet. Can be identified by chromatography or mass spectrophotometry. Another one is Homocystinuria. This is an intermediate amino acid issue with the synthesis of cysteine from methionine. Here we see we can do a Guthrie test as well, but usually the big kicker that you have to know with homocystinuria, if you have too much homocysteine, you can have a thromboembolic event like a stroke or a heart attack before age 30. So they do end up with some blood clotting issues and can have throat clots. So that is what you want to know with the homocystinuria. Two others that there is an inherited enzyme deficiency in the urea cycle is Argininosuccinic aciduria and Citrullinemia. Just be familiar with those as an amino acidopathy. The last one here is cystinuria. This is a defect in the amino acid transport system and not a metabolic enzyme deficiency. So this one you're not missing an enzyme, but it's an error in transport. We usually see a 20 to 30-fold increase in the urine cysteine, so we do see cysteine crystals, and we may see defects with the kidneys as well. The excretion of lysine, arginine, ornithine, and cysteine are measurable, but cysteine is a troublemaker. We do see kidney stones or urinary calculi. I do have a video here with somebody talking about cystinuria and what that means if you'd like to go back and watch that at your leisure. But again, cystinuria, transport, my students say, well, car starts with a C, so does cystinuria. This goes back a little bit to how we diagnose these. Well, usually, if the child is sick, hopefully they don't get to that point because they will have been screened for it right after birth. But let's say they're not. Parents refuse to have the screening done, which can happen sometimes. We do find that the symptoms are nonspecific. They stop eating. They have a failure to thrive type situation, persistent vomiting, liver abnormalities, etc. But in most states, there is a neonatal screening that we can usually find the issue within that. We can also have a prenatal diagnosis, which is when the mother has previously given birth to a child with a severe inherited defect. They may check ahead of time to see if they can treat that baby right at birth rather than waiting till the testing comes back. Here's some of the testing that we can do for this. They use things like thin layer chromatography and the Guthrie test. We can also use capillary electrophoresis, gas chromatography, high performance liquid chromatography, ion exchange. You don't have to memorize all of these, just be a little bit familiar with the Guthrie test for PKU, some of the chromatography for the amino acids, and we can get into some more molecular genetic testing as well. right, so now we're done with the small little amino acids. We're going to start looking at the huge building blocks of proteins. So we have more than 40 of those amino acids in a row. Everything has successfully happened. All of the amino acids have lined up and made a protein. We have thousands of different proteins in the human body. This is responsible for 50 to 70% of the cell's dry weight. Structural elements of cells and tissues are what proteins, where they go. They're soluble in intracellular and extracellular fluids. They're routinely analyzed in the laboratory. We test for so many of these. We find them everywhere, in the blood, the urine, spinal fluid, amniotic fluid, saliva, feces, and peritoneal or pleural fluids as well. So what you need to know about proteins, they're linked via peptide bonds. So whenever you think peptide bonds, think amino acids and proteins. Very short chains can be called dipeptides, tripeptides, tetrapeptides, pentapeptides. So obviously di would be 2, tri is 3, 4 is tetra. If you have more than 5, we call it an oligopeptide. And longer chains can be polypeptides, greater than 40 is proteins. It's the third time we've talked about that now today. We can have different structures. They can be fibrous, which if you think about it are long fibers. Okay, those are manually structural, things like fibrinogen, troponin, collagen, and myosin. Or they can be globular, which as you think of globs of things. Hemoglobin has glob in it. Peptide hormones, plasma proteins, etc. They're very compact. They can denature or fall apart very quickly. So there's a couple different types. This you need to be very familiar with. These are the 4 levels of structure. What we have first is the primary level of structure. When the primary structure when building a protein is the amino acid sequence. So we have that here. All of the amino acids are lined up in a row in a big string. Okay? So they're all just lined up. Boom, 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 one after the other. Then they start to do a secondary level of structure, which is a reoccurring arrangement of the primary structure in one dimension. We call it an alpha helix or a beta pleated sheet. Kind of starts to look like DNA. Okay, so they start to fold in on each other. The tertiary is intermolecular folding. So now not only do we have them in a beta pleated sheet, they're all folding in on each other. It involves the intermolecular folding of the amino acid chain into a 3-dimensional structure. Then the last is quaternary. This is several polypeptide chains in one aggregate unit to make that solid protein. So those are kind of the 4 steps or 4 levels of structure per se. You would need to be able to recognize the difference in, for example, a multiple choice test. So what are the properties of these proteins? Well, we usually separate them and identify them based on some certain properties. The molecular size, which is, obviously how big or small they are. Their differential solubility. So what do they do in a liquid? Okay, are they soluble? Do they float? Do they sink? There's different things that can happen. Their electrical charge. Some have really strong electrical charges. Some of them are just very weak. The adsorption on finely divided inert materials. So how do they react with different materials? And their specific binding properties to antibodies, enzymes, and hormone receptors. So there's different ways that we can figure out what proteins are which. They also have certain functions within the body. They are enzymes that catalyze biochemical reactions, so all enzymes are made out of protein. Proteins, polypeptides, and oligopeptide hormones help regulate your metabolism. Proteins are also antibodies in the complement system, which protect against infection. Plasma proteins maintain the osmotic pressure of your plasma, which is very important, because otherwise you'd blow up like a balloon, or you would deflate. Transport hormones, they transport hormones, vitamins, metals, and drugs. We've got apolipoproteins that solubilize lipids, which you're going to find in future chapters is very important. Hemoglobin carries oxygen, something that we did in lab last week. You may have used either the stat site or the, what's the other one I'm trying to think of? The HemoQ. Protein coagulation factors affect hemostasis. So when you cut yourself, you need coagulation factors to stop yourself from bleeding. Those are proteins as well. So as you can see from this long list, and I don't need you to memorize this whole list right here, but everything is made out of protein, it seems. So as you saw, there's hundreds of them present in the blood. They're made by the liver. Include things like acute-phase proteins, carrier proteins, fibrinogen, other factors, complement, immunoglobulins, enzyme inhibitors, precursors. Don't have to memorize that list, but it's pretty much the same list that we just had on the last slide. In a sense, most things are made out of protein. So let's go over each of the different types of proteins here that you're going to need to know. But one of the first things I'm going to talk about is an acute phase reaction. An acute phase reaction is a nonspecific response to inflammation or tissue damage. This is such a broad thing to have happen. An acute phase reaction can happen when I kick you in the shin. An acute phase reaction can happen when you get in a car accident. An acute phase reaction can happen when you have a heart attack. It is a pile of inflammation that happens somewhere in your body that causes the release of certain things. C-reactive protein would be one of them. We'll talk about that coming up in a couple slides. But the first protein I want to talk about is albumin. He is the most abundant in the plasma. He's synthesized in the liver by the hepatic parenchymal cells, and his primary function is to maintain osmotic pressure. So you don't want to have too much, you don't want to have too little. He binds and transports a large number of compounds. Usually, he's only increased with dehydration. We don't see that albumin level increased in a whole lot of things other than dehydration. Although we do see decreased levels in a lot of cases. Albuminemia, which is a rare deficiency, having inflammation can cause a low albumin. Having liver disorders can cause a low albumin. Renal tubular damage can cause you to urinate out all of your albumin. Gastrointestinal issues, protein malnutrition, not eating enough protein, and having edema or ascites. Edema is having excess fluid in the tissues. Ascites is excess fluid around the liver. When we talk about albumin, we also talk about globulins on the other side of that. Every patient has a total protein done. Well, not everyone, but a lot of them do when they get their blood drawn for an albumin. They will also get a protein. I was going to try and draw something here, but it's not going to work for me, so we'll just get rid of that. All right, so let's say your total protein level is 8. Okay, so you do total protein. Total protein equals albumin plus globulins. Okay, so if your total protein is 8 and your albumin is 3.5, that means that your globulin would be 8 minus 3.5, or 4.5. So your 4.5 would be your globulins. Total protein equals albumin plus all of the globulins, which is pretty much everything else. In some cases, a doctor may want to do an albumin-globulin ratio. That is the ratio of albumin present in relation to all the globulins. This ratio can be interpreted in concentration, or usually it's 1 to 1. All right, so here are some things that can happen. If the globulin is increased disproportionately to albumin, resulting in a decrease in the albumin-globulin ratio, it could be caused from inflammatory issues such as ulcerative colitis. Neoplasms or cancers could be another issue. We want the albumin-globulin ratio to be about 1.1 to 2.5. So you don't want it to be, anywhere too low. As you can see, this one right here, the ratio is 0.778, which is slightly below that reference range. The next protein is alpha-1 antitrypsin, sometimes called A1A. This can be involved with, it inactivates serine proteases, which are used with blood clotting in the immune system and inflammation. It's synthesized in the liver. It inhibits leukocyte elastase, which is released during phagocytosis, which is unfortunate because obviously we want phagocytosis to happen in the right scenarios. It's decreased with neonatal respiratory distress, pancreatitis, and protein-losing disorders. But usually we see it present in high levels in emphysema patients. Also, it can be decreased with liver disease, but emphysema is where I tend to see that associated with. The next one is alpha-1 fetoprotein. This is present in the embryo all the time. It's synthesized in the yolk sac. But it can reappear in the adult during pathological states. A couple things can happen. It can be elevated in the maternal serum, so if you're pregnant and you get your blood drawn. If you have high alpha-1 fetoprotein, it could mean a neural tube defect in the fetus. Or it's increased in adults in hepatocellular carcinoma. So alpha-1 fetoprotein we see increase with hepatocellular carcinoma. Ceruloplasmin is another type of protein. This one carries copper. We need copper to obviously travel from different parts of the body to the other. It's involved in plasma reduction and oxidation reactions, and it's important in regulating the ionic state of iron as well. So copper is very important. Well, we can't just let copper float around by itself. He's carried by ceruloplasmin. If you have a deficiency of ceruloplasmin, you may have some other things that happen. You could have a copper insufficiency, obviously. Inability to release copper from the GI epithelium. You may have an inability to insert copper into developing ceruloplasmin molecules. Or it could be from blood loss, GI renal protein losing syndromes, or something called Wilson's disease. I'm going to talk about Wilson's disease here. Wilson's disease is where the total body copper is increased so significantly that it is deposited in the tissues. And I put this here because we talk about ceruloplasmin, so I kind of talk about copper. But this results in Kaiser-Fleischer rings, or a brown ring around the cornea of the eye. You can kind of see me pointing at it here. That copper buildup can cause damage of the kidneys, brain, and the eyes. If you remember Home Improvement with Tim Allen in it, his neighbor Wilson, when he'd peek over the fence, all you could see is his eyes. So Wilson's disease, think about those eyes. Here's that C-reactive protein that I was referring to. This is one of the first to become elevated in inflammation. Remember when I talked about the acute phase reactions? Any type of inflammation that's occurring in the body, a C-reactive protein would be increased. So if you had a heart attack, C-reactive protein would be increased. If you got in a massive car accident, C-reactive protein would be increased. Anything that would cause inflammation. My son, after he was born, he was in the NICU for 5 days because he had a high C-reactive protein, which irritated the daylights out of me because I know that test doesn't really tell you anything. It doesn't tell you what's wrong with the patient. It just tells you that something's inflamed somewhere in the body. So the only use that it's good for is to monitor the course of treatment in a disease. So once my son was born, they figured maybe he had an infection. So they gave him antibiotics, and then they drew his CRP level every day, and to make sure that that CRP level was coming down. So you could kind of monitor to make sure that whatever the inflammation is that it's going away. We find it increased with, like I said, heart attack, stress, trauma, infections, whatever. It's good for screening and assessing inflammation. Definitely not good for diagnostic purposes. Next one is haptoglobin. This one binds hemoglobin irreversibly, so it cannot be excreted by the kidneys. Haptoglobin hemoglobin complexes are also important for the control of local inflammatory processes. It's increased by corticosteroid hormones and NSAIDs, which are like ibuprofen. A decrease indicates intravascular hemolysis is going on. Estrogens can decrease it, and liver disease can decrease it as well. But its claim to fame is carrying hemoglobin. Retinol-binding protein helps carry vitamin A. We find this increased with renal disease and metal poisoning, decreased with liver disease and malnutrition. Transferrin, when you think ferrin, think iron, is the principal plasma transport for iron. It's interesting to know that in iron deficiency anemia, transferrin levels are increased. You'd think that if iron is high, that transferrin would be high, but these are opposite. So if transferrin is high, iron is low. The next one is called Transthyretin or Prealbumin. This transports some thyroid hormone in the plasma. It's also increased in Hodgkin's disease and glucocorticosteroid therapy, but we see decreased levels with poor nutrition. So if somebody's on tube feeding per se, and they want to make sure that the body is able to absorb that protein and actually use it, they might do a prealbumin to check and see if that tube feeding is working appropriately. Another type of protein are complement proteins. You're going to learn a lot about this in your immunology class in a couple quarters, but there's 20 proteins in the blood and tissues that work to destroy viruses, bacteria, or sometimes your own cells. There's different types of pathways involved in these complement proteins and how they destroy foreign organisms, but we're not going to go into detail here. Just know that the complement proteins destroy foreign invaders in the body. These are synthesized in the liver, just like all your other proteins are. Immunoglobulins, these are antibodies, humoral antibodies. Humoral just means that they're in your plasma. They recognize foreign antigens or foreign things that may come in your body, foreign invaders, and attach to them so complement can come in and help destroy them. So when we refer to having protein abnormalities, we may say they have hypoproteinemia or hyperproteinemia. Hypoproteinemia is usually when they're less than the reference range. It occurs when there's a negative nitrogen balance. Sometimes we refer to protein as nitrogen balance. It can be caused by an excessive loss, decreased intake, decreased synthesis, or some type of breakdown. Hyperproteinemia is a total increase. A big one is dehydration. Remember that albumin is increased in dehydration? When the concentration of proteins is elevated due to decreased volume of solvent water sometimes, or excessive production, which could be something like, don't memorize this, but Waldenstrom's macroglobulinemia, multiple myeloma, those have excess production of proteins. We'll talk about those in immunology, but those are some diseases that can just produce too much protein. Some of the methods that we can do a total nitrogen, which assesses nitrogen balance, which gives us a good indication of how much protein there is. The reference interval for proteins, total protein, is 6.5 to 8.3 grams per deciliter. And the measurement is the Kaldal reaction usually. This is how you say that, Kaldal. Another thing that we can do, which we did in lab 2 weeks ago, was serum protein electrophoresis. We did it with color, but this is done with proteins. Usually we take the patient sample and we can run it through a gel, just like we did with our colors, and they're applied to close to the cathode and they travel towards the anode, usually at a pH of 8.4. Once they travel through, we can put a dye on the gel. It'll dye the proteins in the gel, and we can run it through something called a densitometer, which can tell how much protein is in that spot. Here's what it would end up looking like. So if we had each patient sample, patient 1, patient 2, patient 3, patient 4, what happens is you apply electrical charge, it travels through the gel, and you can see the different bands here. Okay, so those are areas where as far as it could travel through the gel. And you'd be able to run it through a densitometer. The darker the line, the more protein that would be there. Other methods of analysis: high-resolution protein electrophoresis, just a little fancier, capillary electrophoresis, isoelectric focusing, and immunochemical types of methods are just something a little bit different that can be done. Other body fluids that we test proteins for are your urine and your spinal fluid. These are really important. You should not have a lot of protein in your urine, if not really have any. What this could mean is that you've got renal tubular issues, or you've got a breakdown in your glomerulus where protein are supposed to be repelled, but if they're making it through in your urine, you could have an issue there. In spinal fluid, sometimes if you've got too much protein, it could mean that you have a bacterial, viral, or fungal meningitis going on. You could have multiple sclerosis, some type of neoplasm, a disc herniation. There's a lot of things that can cause too much protein in a spinal fluid, but we can take that spinal fluid and tell the physician exactly how much is there. And that concludes your session on amino acids and proteins. Thank you.